you see with the flow cytometer. And then you get a lot of data, and then you use various high-dimensional reduction analysis approach. So we did that. I will just show one example in that view is, so what this shows is the data coming from 8 tissues of a mice. And I think you will appreciate that even, for example, dendritic cells, which are coming from different tissues of the mice, they are different. So this is a 3D projection coming from staining of a cell with 37 markers. So depending on the expression of those 37 markers, now you can distinguish different cell subset coming from different tissues. We did the same thing with the lung myeloid cells. And of course, when you do this, you find there are various cell subsets in the lung. For example, 4 cell subsets of neutrophils, so many cell subsets of DCs. And what she found is that upon infection, some of the myeloid cells, so these are monocytes, which comes up during the infection. And when these animals are being treated, this goes off. So, and it is almost similar to what happens in an uninfected mice. So we went on to show which cell subset is important. And the overall summary of this is that upon MTB infection, there is reduced sirtuin-1 expression and its activity. And now if you can enhance the sirtuin activity by activators, either synthetic or natural, you can reduce the bacterial replication. So this was in mice, what we did. We continue with the monkey model. So what we did, we took monkeys, infected them with a dose by which you know that monkey will develop an active disease. So 1,000 CFU, you either leave them, no treatment. You either treat them with one regime here, pyrazinamide, moxi, and ethinomide, and in one of the group, we added pterostilbene, which is again a synthetic, natural activator of sirtuin-1. And what we found that the monkeys which received pterostilbene as an adjunct to the anti-TB therapy, they have reduced bacillary load in the lung and reduced pathology. We are continuing these experiments, adding more monkeys to it. However, if I want to summarize my work here is what we have shown that there is a deep crosstalk going on between AMPK and sirtuin-1, and both regulates each other. And now if you activate these metabolic sensors by different activators like metformin, resveratrol, or other boosters, you can enhance the mitochondrial function, oxidative metabolism, and other innate immune response mechanisms. And together, this will have an effect on protective and pathologic immunity. With that, I would like to stop. I would like to thank the people who have conducted a lot of work. Catherine, Julia, these are 2 postdocs in the lab. Now Dilip has joined with us. Various collaborators in the institute, in A-STAR, and from east to west of the globe, and the funding agencies. And now we have started working on gram-negatives also, and we are organizing the first AMR symposia in India, so you are welcome to join. Thank you.
Thank you, Amit. Very nice talk. So we have time for 1 or 2 questions.
Hi, Amit, nice question. Nice talk. So my question is regarding, your sirtuin activation, you are checking the monocytes. So what is the dynamics of looking at activation of these molecules in circulation versus, because the infection is localized,
Yes.
in the microenvironment.
Yes.
So how is the dynamics work between peripheral tissue,
We have looked at the sirtuin-1 expression and activity in a mice, in the lung, and in blood. So expression, we didn't find reduced expression in the blood. We did find in the lung. But activity, we found reduced activity in the lung as well as in the blood. In monkeys, we didn't look in the blood, in the periphery, but we look in the lung. So our case is more of a localized. So whenever we try to do something which could reproduce the infection site.
And in the granuloma tissues, Are sirtuins localized to CD14 or 11 markers or CD68
So
markers?
they are, so it's been expressed a lot by CD68 or CD163 positive macrophages.
So that's the macrophages.
Macrophages, but it is also there in T cells.
In T cells.
In T cells, yes. So we have something else on the T cells part, which I have not shown, that is the work in progress.
Okay, thank you.
So you have shown that SIRT1 was overexpressed under latency conditions, right?
Yes. I won't say it's overexpressed, but the expression doesn't come down compared to the
Right.
normal individuals.
So it's not a latency antigen.
No, it's not reduced.
Right. Okay. Any more questions? Yes.
So you said that metformin may have an antibacterial activity in this case. So any evaluation that if it starts having resistance against metformin, then especially in a country like India where metformin is widely used, so what in that condition, if it will definitely aid to the spread of AMR?
Sure. So metformin does not have a direct antibacterial activity against any bacteria. It's only when you treat a host cell or the host. So there is no direct MIC. So that's why there will be no drug resistance against.
How about horizontal gene transmission? Any idea?
Horizontal gene transmission?
Yes, like if there is some genes that acquire resistance against metformin, and while conjugation or transduction, it can be transmitted to the potential infectious pathogen.
That could be a possibility, but for my knowledge, I do not know as yet if there is any gene in any bacteria which shows resistance against.
Sir, I'm not talking about in particular mycobacterium. I'm just asking that if some drug is causing resistance, then horizontal gene transmission can occur to pathogenic or non-pathogenic both. So that can be a mode of communication of AMR. That is my point. Thank you.
Yeah. Thank you.
I think a question over there.
Wonderful talk. I'm not from the field of TB, but I have a basic question. So SAT1 has been known to involved in degeneration and also found highly expressed in hippocampus and neuron. So what do you think that, I'm as a biologist, I'm just a neurobiologist, I'll ask you, if you are targeting these two crosstalks, SAT1 and AMPK, so don't you think you might also modulate some activity in brain? Because in Alzheimer also, IGF-1 was found to increase the SAT1 expression.
Okay. So what I know is that it's a molecule which is being very well studied in aging field. And it enhances the aging. It is associated with the aging. But when we give any of these supplements or drugs which target these metabolic sensors, so you will have an effect everywhere.
True.
And definitely in hippocampus and neurons, you will have an effect. We have not studied that.
So would you like to see in the changes in the other, gene expression status in brain or other?
Yes, definitely. I'll be interested.
Thank you very much.
One last question, just.
Okay.
Curious about, I liked your talk, though I came towards the end. I'm a bit curious about what would you visualize if the physical activity is coming into the picture of your research?
Physical activity.
No activity, mild, moderate, and extreme physical activity in presence of resveratrol.
Okay, that's a challenging question.
In presence and absence of resveratrol.
I can't say anything about that. So I know like caloric restriction enhances the SIRT1 activity. And there is a group now which is studying, so they are studying the effect of intermittent fasting.
Sorry?
Intermittent fasting
Yeah.
on infections, and they are using TB also as one of the model. But for physical activity, I have no idea.
This is just a hypothesis.
Yeah.
As an antioxidant, I think in the moderate and extreme physical activity, the ROS generation would be higher,
Yes, rosination.
right? So in presence of resveratrol, the ROS levels should go
down.
low.
Yes. So there should be a proper balance, what I think. because
That's what I
too
would
much
think. But
ROS is also not good.
yeah,
It
but
can
I
lead to
would be
oxidative-
interested in case
Sure.
there could be collaborative ways of carrying out such kind of research
Sure.
in future. Thank you.
Thanks.
This is Dr. Ishwari.
Okay, thank you.
Thank you, Amit. I think we should move on. I would like to welcome our second speaker, Dr. Baswati Pandit at the National Institute of Biomedical Genomics in Kalyani, West Bengal. And Dr. Baswati is going to talk about the role of host genome variants modulating outcome of TB infection.
Good afternoon, everybody. First of all, I would like to thank the organizers, particularly Dr. Samuel Raj, for giving me this opportunity to talk here and present some of our works. So my talk will be investigating the role of host genome variants and its role in modulating the tuberculosis infection, mainly